Hi everyone, and welcome to our review on DNA replication. So what we're going to be aiming to do in this little summary video is go back over the actual process of semi-conservative DNA replication. Now, as you can see from the spec statement here, then we need to know the roles of a couple of key enzymes. We're actually going to look at four, and we're also needing to understand what this phrase semi-conservative DNA replication actually is. We'll also touch briefly on mutations. Mutations will come up again at later points in the A-level biology course as well. Okay, so before we launch into the process of DNA replication in any detail, let's just remind ourselves why we need DNA to replicate in the first place. Now, one thing that we've looked at in one of our earlier videos is the whole idea of the cell cycle and cell division. Now, in order for a cell to be fully functional, it is going to need DNA there, because that DNA is obviously containing the codes for proteins that we need, etc. So every time that cell is preparing to divide, we need to copy the DNA so that every new cell will have that DNA present. And that's going to occur during the part of our cell cycle called interphase, if we remember. One thing that we should remember is when we're considering our DNA, remember, it is this very well-known, anyway, I hope it's well-known, shape of a double helix. So we've got two strands there. Now, what actually happens in DNA replication is that each of those strands in our original double helix is going to act as a template for creating a new double helix. When it comes to our DNA replication, we're going to learn about four key enzymes here: gyrase, DNA helicase, DNA polymerase, and ligase. Now, what we're going to do is we're going to take each one of these in turn as we go through the process of DNA replication to actually see what they do. First thing that has to happen in terms of our DNA replicating is when we're starting with our little double helix, we need to separate those two strands. So that's a process we refer to as unwinding. So the DNA unwinds, and the unwinding is achieved by the gyrase enzyme. So basically what we're doing here is we're going to open up our DNA. Okay, so it's going to unwind, and that's the gyrase. Next, we are going to unzip that DNA strand, because what we hopefully remember is that running down between these two strands, then what we've obviously got are the little bases. So there's one of our little sets of bases, and then on the other side we've got the other bases, and between them we've got hydrogen bonds. Now what we've got to do is we've got to unzip this by breaking the hydrogen bonds. Now that breaking of the hydrogen bonds is carried out by our DNA helicase. So gyrase unwinds, DNA helicase breaks the hydrogen bonds between the nucleotide bases. Once we've actually got our DNA all nicely exposed, so we've got our little bases running down each side, then what we are able to do is create some new complementary base pairs. The reason we can do that is that in the nucleoplasm, so that's obviously within the nucleus, we have got 3 phosphorylated nucleotides. So remember, our nucleotides are going to basically end up as our A's, T's, C's and G's. Now what's going to happen is that where we've got our A, then one of our 3 phosphorylated nucleotides is going to come in, and we're then going to join onto there. So that's going to obviously follow down the strand in order to actually make the new ones. The way that these nucleotides are joined together is through the formation of a very special type of bond, phosphodiester bonds, okay? And this is going to create this thing we refer to as the sugar phosphate backbone. Now, in order to create this sugar phosphate backbone, if we think about the basics, let me just try and do a quick sketch, then what we actually have is something along this line. So, that's terrible. But we've got our little phosphate group joining our 2 sugars together. So this is going to be our sugar phosphate backbone. Now, what we have is these two bonds here are going to be ester bonds, and because they're between a phosphate group and there's two of them, then we have a phosphodiester bond. So this is our sugar phosphate backbone that runs up, and then obviously on the inside here we would have our little bases. So that is DNA polymerase that achieves this. So DNA polymerase, our third enzyme, that creates phosphodiester bonds in order to make the sugar phosphate backbone by joining nucleotides together. One thing that you should note is that when we're looking at the direction in which this occurs, it runs in the 5' to 3' direction. So basically on our strands, what we have is we have got our 5' end and our 3' end. On the other strand, because this is anti-parallel, then they would be the other way around like this when they're joined together. But synthesis is always 5' to 3'. So remember that fact. Now, obviously, common sense might be telling you, if we're separating this out a little bit at a time like a zipper, and then we're having this synthesis taking place, this joining of our free phosphorylated nucleotides onto those strands, how does that work going 5' to 3'? Well, we can call these two strands different things. We've got the leading strand and we have the lagging strand. Now, the leading strand is always going to be synthesized continuously. So that literally means that we start at one end with our little primer, we just go all the way down, just adding new little nucleotides right the way down. No gaps, just a nice continuous strand. The lagging strand is obviously different because it's running in the opposite direction. So what we find is it starts synthesizing closest to the little join of the zipper, if you like. So if you think this is our little joining point of our zipper there, then we need to start synthesizing down at this point. So we can't just make one whole strand, because obviously we're having to make it a little bit at a time as this opens up further. The way that this works then is we're going to basically make little sections, and these are called Okazaki fragments. So what we do is we make one of our little Okazaki fragments here, then as it unzips a bit further we have another Okazaki fragment made here, and then later on we're going to join those two fragments together with our fourth and final enzyme, the ligase. Because remember, ligase, if you think what ligate means, that's all to do with joining. So ligase is going to be the enzyme that joins those short fragments together in order to make our complete strand. Now, what we're actually going to need is energy to achieve this process. Things don't just join together by magic, they actually need to form some kind of bond. How does this happen? Well, if we think about what we said earlier, we have these phosphorylated nucleotides. So if we hydrolyze, which is obviously where we're going to carry out our hydrolysis reaction, and hydrolysis is splitting because lysis just means splitting, then what we are going to do is release these extra phosphate groups. Now, in doing this, we are going to then provide the energy needed to make those phosphodiester bonds between the sugar of one nucleotide and the phosphate of the next. So quite simply, by hydrolyzing the phosphate groups off those phosphorylated nucleotides, we get the energy needed to then create those phosphodiester bonds. Now, obviously at the start of this, we mentioned the fact that this is called semi-conservative replication. What does that mean? Well, if we're thinking about the actual words, okay, so semi is usually half, okay, so that's how we've heard of it, like semicircle, half a circle, we know this. And then if something is conservative, or we're conserving something, then basically that's like keeping, okay. So if you think of semi-conservative replication as keeping half the original, that's what we mean. So that what we actually have, if we start off, if this is our original DNA double helix, my drawing's getting worse, I do apologise. If that's our original double helix strand, then once we've actually made our new strands, we are going to have one is going to be the original, the red bits, and then the other strand is going to be the new one, which is the blue. So what we've got, half original and half new. Now this does come up occasionally, asking you to explain what's meant by semi-conservative replication, and what they're looking for is that it has one original DNA strand and one new DNA strand. So just make sure that you put both parts of that argument in. It is worth remembering that when we're looking at this process of DNA replication, it's not just occurring in eukaryotic cells' nuclei. We also have our prokaryotes and two structures within our eukaryotic cells, mitochondria and chloroplasts, that have their own DNA. So when we're undergoing DNA replication, we need to replicate in the mitochondria and the chloroplasts as well. Just like in prokaryotic cells, they need to replicate their DNA. Slightly different in terms of what's happening there. And so what we actually see is, because the DNA is a loop, so remember what we said, our DNA inside our prokaryotes, mitochondria, chloroplasts occurs as a loop, then it's not going to be a case of unzipping and so forth. Instead, we end up with a little bubble sprouting from our DNA loop. So basically we end up with this little bit here like so that kind of sprouts out, and from that it's then going to unwind and unzip. Now what that means is that we then have these complementary nucleotides able to join to the exposed ones, and then we can replicate from there. Now in terms of how this semi-conservative replication idea came about, as always in science, it wasn't a case of someone sat there one day and just said, this is it. There were different theories proposed. In fact, there were 3 different ideas on how DNA would replicate. So if we have a look at each in turn, we'll then have a look to see how we proved semi-conservative. Option 1 is what we refer to as conservative replication. Now, in our conservative replication, basically what we're starting off with is our double helix, as always. So there's our standard double helix that we start with. The original DNA acts as a template, but when we make our new DNA, we are still going to have one double helix which is completely the original. The second double helix is going to be all new. So conservative replication, we've got one original and one entirely new double helix. So that was conservative, that was one idea. Option 2, as we've already discussed, semi-conservative, we have our original DNA strand acting as our template, and then when we've actually replicated it, what we have, and I do apologise for the drawings of the double helixes here, they are getting a bit scrappy, is you would have one original and one new. So that gives us semi-conservative, one original strand, one new strand in each case. Option 3 is possibly the strangest one to get your head around of all, something called dispersive. Now this one is basically where, when we've actually made our final strands, I want to try and draw this, it's going to be interesting, then what we end up with is these little sections that are going to be new, and on that same strand we've got the original. Oh dear, it gives you the idea. So basically what we end up with are 2 strands, I'm just going to put ×2 because I don't want to try and draw that twice, 2 strands where we've got some original fragments, some old fragments, but they're all on the same thing. So how did we actually prove that it's semi-conservative? Because it is, that's what we now can prove. We know that it's semi-conservative replication. How did we get to that? Well, it was a set of experiments carried out by these guys called Michelson and Stahl. Now, what they actually did here was they grew E. coli in what we refer to as heavy nitrogen. Now, the heavy nitrogen is basically just an isotope of nitrogen that obviously has a higher mass number. So they grew it like that, so the DNA of our original strand contains the heavy nitrogen, and then they grew it for another division in the light nitrogen isotope. So basically what they could then do is, by centrifuging the results of this, we could then get bands based on the mass of the DNA. So what do we find? Well, in the conservative replication, we would have expected there to be two distinct bands. Because if we're saying that this is going to be made up of one completely original and one completely new, then the original one is going to be the heavy nitrogen and the new one is going to be the light nitrogen. So we end up with two bands, one for each. If it was semi-conservative replication, then what we're saying is, because both of them have original and new strands on there, that means that these are going to be the same mass. So we have a single band in that tube for semi-conservative. If it was dispersive, then depending on which bits of the molecule came from the original and which bits were the new, then we're going to end up with a whole array of little bands. So basically we just get a whole mass of different bands in the middle there. When they did their experiment, they absolutely found that they had a single band. Therefore, that supported semi-conservative, because all of those DNA bands that they had were the same mass. Last thing we need to cover is just this idea of mutation. And hopefully from our GCSE, we do remember that when we say mutation, we are talking about a change in the DNA sequence. Now, why does this affect things? Well, if we have a mutation, so a change in that DNA sequence, then what we're going to end up doing is changing that triplet codon. So remember, when we're looking at our DNA, then what we end up with is just a whole string of our bases. Now, when we actually come to make a protein, going back to some of our earlier work on protein synthesis, these are read in sequences of 3 bases, those triplet codons. Now, if we've got our original strand like this, obviously our first triplet would be TAA, next one CGG, then CAT, etc. If we were to have a situation where we had a mutation and therefore one of these changes, so if we were to then change that sequence to, let's just go TAT, CGG, CAT, okay, then we've obviously changed this codon. No longer is it TAA, it's now TAT. So, what do we know about these codons? They code for an amino acid. That means that because we've got a different DNA-based sequence, we've got different triplet codons, therefore, potentially, different amino acids would enter the primary structure of our protein. Link back to our biomolecules work, primary structure of protein is the order of amino acids. Now, if we get a different sequence of amino acids, then we can see different secondary or tertiary structures if the side chains have different properties. So because of that, we get a change in that 3-dimensional shape of our protein. And if we're talking about an enzyme, for example, this is a problem, because if we change the 3D shape of our protein, we've changed the shape of the active site. Therefore, it's no longer complementary to the substrate. It won't work in that reaction anymore. So this gives you one little idea of how a mutation could lead to problems in terms of organism function. How often do mutations occur? Well, they occur in approximately 1 in 10 to the power 8 base pairs. So they're not massively common, but they're also not rare. Now, the good thing is that because our work on the cell cycles tells us this, then we know that there are checking mechanisms in place. So most of these mutations are going to be identified in our proofreading parts of the DNA replication sequence and therefore edited out, so they won't cause problems. Obviously, that is not perfect. There will always be some mutations that get through, hence why we have cancer to this day. As always, I hope you found this video useful. Don't forget there are additional resources over on the website that you can utilize to hopefully help you with your revision. And don't forget to subscribe so you can see when I post the next video.